exocrine secretion and an endocrine secretion. We really talked about the endocrine portion when we did the endocrine system. We talked about insulin and glucagon and a little bit of somatostatin, but mostly insulin and glucagon, so we won't revisit that. But the exocrine portion is primarily two major components. A bicarbonate, HCO3 minus fraction, which neutralizes the acid coming from the stomach. Remember, these exocrine secretions are being secreted into the duodenum, the first part of the small intestine. So we neutralize that acid. That's going to raise the pH, make it more alkaline, isn't it? So we want to do that. And the other thing is the enzymatic fraction. We mentioned yesterday that the enzymes in the small intestine come from two major sources. One is right there in the small intestine. There are some enzymes that are made and they're attached to the microvilli. We call that the brush border, so they're called brush border enzymes. The other major source of digestive enzymes in the small intestine is from the pancreas. So those are the two exocrine secretions, digestive enzymes and bicarbonate ions. We produce a number of different enzymes that go into the small intestine from the pancreas. Some of these are proteolytic, digest protein. Some of them are going to digest polysaccharides, carbohydrates. We call this pancreatic amylase. It's the same as the amylase found in the starch, but because it comes from the pancreas and not in the saliva, we call it pancreatic amylase. And then we have some enzymes that are going to digest the lipids, the fats. This is lipase, pancreatic lipase. And finally, we have some that are going to digest the nucleic acids. The proteolytic enzymes, you recall, must be produced and secreted in the form of a precursor, a zymogen as they're also called. And when they get into the lumen, we have one of these proteolytic enzymes called trypsinogen. Now that's a precursor. And we have other precursor proteolytic enzymes. They come into the lumen of the small intestine. And one of our brush border enzymes that are attached to the microvilli that are made locally, a enzyme called enterokinase. There's the name there with the arrow. Enterokinase converts trypsinogen to the active form, trypsin. And trypsin converts all the other inactive proteolytic enzymes, zymogens, into their active form. So now they're in their lumen and they're all activated and they can start digesting the proteins. So that's what's happening there. Now this table may make you feel a little overwhelmed, but basically this lists the major hormones in the digestive that act in digestion. And first of all, let me reassure you, you're not going to have an essay question on this. But we might have some multiple choice or multiple choice questions. And that would predominantly be looking at the hormone and its activity, the function. So the hormone way over to the left side and its activity over here. So we have cholecystokinin and then its functions over here and all of that. So know it to the level where you can recognize if you're needing to sort of match up the functions with the hormone. somatostatin, for instance, just to help you, that's pretty much inhibitory on everything that it does. So that's, we know that that's going to have inhibitory actions. Secretin, as we've already mentioned, is going to increase the exocrine pancreatic bicarbonate, especially the bicarbonate fraction, whereas cholecystokinin is going to increase the enzyme-rich pancreatic exocrine secretion. So there's some differences there. So you can see that and sort of take note of some of those differences. Before we get into the digestion and absorption actions a little bit more fully, let's chat about some of the regulatory activities of secretion and motility and that kind of thing. And we'll start looking at the stomach, the regulation of the stomach. So gastric function. If we look at gastric motility and secretion, we can separate the phases controlling these things into three main phases: cephalic, gastric, and intestinal. Cephalic and gastric gear up the activity of the stomach, both secretion and motility. The intestinal phase is a negative feedback. It starts tamping it down, reducing it. Cephalic, reference from the brain or the head, is whenever you start to smell food, see food, taste food, it initiates the cephalic. It's going to gear up the stomach secretions and motility in preparation of the arrival of the food. So when you're smelling food and your stomach starts to rumble and that kind of thing, that's your cephalic phase kicking in. The gastric phase is when the food has actually arrived in the stomach. And it's going to gear it up even more. And then the intestinal phase is when the food starts going from the stomach into the duodenum, into the small intestine. And now you want to slow things down a little bit. Because again, the intestine, the duodenum, cannot take too much food at a time. So it has to say, slow down a little bit. So those are the three phases. Let's look at those a little in more detail. Here's some of the phases that we see here. In the cephalic phase, we have some enteric neural activity. This activity is within the wall of the stomach in this case. And it's going to be sending signals to say, let's increase secretion and let's increase motility of the stomach. So it's coming down, and we're then stimulating gastrin secretion from the G cells lining the stomach. That's a hormone, so it's going to go into the blood. Gastric secretion will directly affect the parietal cells to release more hydrochloric acid, and it will also stimulate histamine secretion from the enterochromaffin-like cells, the ECL cells. And histamine will stimulate additional gastric secretion. In addition, we'll get some parasympathetic response. We'll get the vagus involved, cholinergic, releasing acetylcholine. That will also stimulate acid secretion. So the cephalic gets things going. But as the food gets into the stomach and distends the stomach, we also move into the gastric phase, shown down here. luminal distension, the lumen distends, and the formation of some amino acids and peptides also increases the gastrin secretion and the vagal parasympathetic response. So you gear it up even more. Your motility, your churning, your muscle contraction is all enhanced. You're basically making your stomach do its job and gearing it up. This continues, of course, for a while, and eventually you're going to move some of the food into the duodenum through the pyloric sphincter, and that's going to send signals, what we call the intestinal phase. You're going to release somatostatin, and that's going to inhibit acid secretion, and you're going to slow down the motility. So the intestinal phase essentially slows it down. This is a little table that gives us a summary of this. Here's the cephalic phase, parasympathetic nerves, enteric nervous system. It's caused by the sight of food, the smell of food, the taste of food, chewing, and you're going to increase in your hydrochloric acid secretion. You're also going to increase motility and that kind of thing. You're going to have more gastrin secretion and all of that, histamine. When it gets to the gastric phase, you're getting a distension, and you're going to get, again, more hydrochloric acid secretion, more motility, more gastrin, more histamine. And then the intestinal, as your small intestine is distended, you're going to inhibit motility and hydrochloric acid secretion. So this is sort of the phases, control and regulation of the stomach's actions that we see there. Anywhere along the digestive tract, we can have reflexes. These are three of the main ones. Gastroileal reflex. What is this about? If we increase stomach activity, increasing the motility of the stomach and all that. In other words, you're starting the digestive process. Food has arrived in the stomach. You're increasing the secretions and all of that. That will send neuronal signals down to the distal part of the small intestine, the ileum, and it will cause it to increase its motility, muscle contraction. Why do we want this? This is essentially, if we want to be a little bit simplistic, the stomach is saying to the ileum, food kind is coming your way. You better start moving whatever you have in out, because more's coming. Okay? So the ileum is moving stuff into the large intestine, through the ileocecal sphincter into the large intestine, to make room for the stuff that's coming from the stomach. And we call that the gastroileal reflex. Now the opposite of that is the ileogastric reflex. Once the ileum gets too distended, it sends signals, neuronal signals, back to the stomach, and it says, whoa, slow down. I'm full. Don't send me anything more for a little while. So the ileogastric reflex decreases the gastric motility for a while, allowing time for the ileum to empty a bit before it says, okay, now you can send me some more. Finally, anywhere in the small intestine and the large intestine, you can have what's called the intestinal reflex. If you get an overdistension in one segment, it will cause relaxation in other parts. So it's basically saying, I'm too full here. Don't send me as much. Relax a little bit. Let me empty a little bit until I can give room. So that's the intestinal reflex. This is mostly, all these reflexes are mostly done by the enteric nervous system, that inherent, extrinsic nervous system. So they're sending signals back and forth. And that keeps the movement through the digestive system in proper balance. Secretion of bile. First of all, we know that bile is made in the liver by the hepatocytes. It is released into the bile canaliculi, and it goes to the bile ductules, and then eventually it goes from the liver. Some of it goes to a little bypass to the gallbladder to be stored. Some of it goes directly into the duodenum. The stored bile in the gallbladder is waiting for a meal, especially a meal high in fats. When you have a meal high in fats, that gallbladder will contract and squirt a bunch of stored bile into the duodenum to help us. Well, what does bile do? Well, bile really has two major components. Bile has bile pigments and bile salts. The bile pigments are things like bilirubin and biliverdin. One is green and one is yellow, and they mix together to form this ugly color of bile. It doesn't look very good. Part of these bile pigments come from the recycling of the breakdown of hemoglobin from the red blood cells when we destroy them. Part of the color also that goes in the feces is due to the bile pigments. Because bile is made in the liver, if the liver is not working appropriately, then it doesn't metabolize the bile pigments correctly, and we get jaundice. Jaundice is when the sclera, the white part of your eye, turns yellow, or your skin turns a little yellowish. And that's telling us that there is some problem with the liver. The liver isn't doing its job. And so if you have a premature infant, a preterm baby born, it might be a little jaundiced because the liver has not reached full functionality. So it's not doing its job. So you might have to put him under a Billy light to try to help metabolize those bile pigments. If you're older, a person might have cirrhosis of the liver, they might have liver cancer, they might have hepatitis, viral hepatitis, or some inflammation in the liver. They could have a number of different liver problems that was then causing this incomplete metabolism of the bile pigments, and therefore the jaundice as a symptom of that. On the other component, and we have water and a bunch of other stuff in here, but these are the two main components of bile. The other are the bile salts. The bile salts are amphipathic, and you remember that means they're partially polar and partially non-polar. And we're going to see later on today the function of the bile salts, these amphipathic bile salts, is to do what we call emulsification of fat. Emulsification is not digestion. Digestion, you're going to break bonds. Emulsification is simply taking big fat globules and separating them into little fat droplets so that they can have more surface area to facilitate digestion with an enzyme. We're going to come back to that a little bit later today, but that's what the bile salts do, emulsification. when we're looking at what controls the secretion of pancreatic juice and bile, we'll just remind ourselves that the hormone secretin is going to be stimulated to be released when the duodenal pH is less than 4.5. In other words, the acidic chyme coming from the stomach into the duodenum is making the pH lower, more acidic, and that will stimulate secretin from the intestinal cells to go into the blood and cause the pancreatic bicarbonate fraction to be secreted into the duodenum to help neutralize this, to help raise the pH. So that's what it's doing. It also puts bicarbonate into the bile. Cholecystokinin, which is also released from cells in the small intestine, is especially secreted in response to fat and protein in the lumen. It acts as a permissive role in secretin, and vice versa. Secretin acts as a permissive role in cholecystokinin. But cholecystokinin stimulates not only the enzyme fraction from the pancreas, but it stimulates the contraction of the gallbladder so that bile is released from the gallbladder and put into the duodenum. It also relaxes the opening, the sphincter of Oddi in the duodenum to allow passage of the bile into the duodenum. So cholecystokinin really facilitates bile secretion and gallbladder contraction. So here it is, cholecystokinin secretion contracts the bile, relaxes the sphincter, bile can go into the duodenum. Secretin, by the way, was the first hormone ever discovered back in the late 1800s by two medical students. Isn't that remarkable that we didn't even know, hadn't identified any hormones until the late 1800s? Well, that's a long time ago, but still, that's a lot of history before we didn't know anything about hormones. Anyway, secretin, they named it secretin, which was a good name, and that's the name of it. All right, well, let's talk about now digestion and absorption. First, we'll talk about carbohydrate digestion, and we'll talk about protein digestion and absorption, and we'll talk about fat digestion and absorption. We won't do much with nucleic acids because that's a relatively small amount. Well, carbohydrates, we have polysaccharides like starches. Then we have disaccharides, which are two sugar units connected by a glycosidic bond. And then we have monosaccharides, right? What we need to do is take any of our polysaccharides and digest them all the way down to monosaccharides, and take any of our disaccharides and digest them all the way down to monosaccharides. We then can absorb the monosaccharides into the blood. All right, so how are we going to accomplish this? Well, the polysaccharide starch in particular is going to be, we're going to initiate starch digestion in the mouth through the salivary amylase. But that's relatively limited. Then it's sort of on hold as we go through the stomach. And when we get into the small intestine, we have pancreatic amylase from the pancreas, which continues to digest it down. Eventually, we digest it down to disaccharides, or we eat the disaccharides. Disaccharides are things like sucrose, lactose, and maltose, and we need to digest them down into monosaccharides like glucose and fructose and galactose. And then we can absorb them. We're going to digest the disaccharides with disaccharidases, enzymes that digest disaccharides. It's interesting that the enzyme names match the disaccharide name. So disaccharides like sucrose, they end in OSE, and you digest sucrose into its monosaccharides using sucrase, the enzyme, the disaccharidase. Lactose is catalyzed, hydrolyzed with the enzyme lactase, and maltose by maltase. So they match in that way. Cellulose, by the way, is a polysaccharide that we don't digest. So it just goes right on through. And you remember cows have other things, ruminants can do that, but we cannot, for instance. This gives us an example of the hydrolysis of table sugar, sucrose, into its two monosaccharides using the enzyme sucrase and water.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces.
Okay, so what that's saying is sucrase, along with all the other disaccharadases, are brush border enzymes. They're going to be attached to the microvilli. Amylase comes from the pancreas or in the saliva. But the disaccharadases are brush border enzymes. They're attached to the microvilli.
This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
So we have the polysaccharides being digested by pancreatic amylase. And we go down to the disaccharides, which are hydrolyzed, digested all the way to the monosaccharides by the brush border enzymes. And then we have absorption. Now absorption, here's the lumen. Here's our epithelial lining cell, or the luminal cell. And here is the cell, the other side, our intestinal fluid, and that would go into the blood. So how are we going to get from here over to here? Well, a lot of it is driven by sodium co-transport. Here is an active transport of sodium going out, potassium coming in, requiring ATP hydrolysis energy. If we pump the sodium out of this cell, that means we have a low sodium concentration inside the cell. Now we have two carriers. One classification of carriers are the GLUT. We actually have at least 5 subcategories of the GLUT carriers, glucose carriers essentially, but they can carry other monosaccharides. The glucose carriers are going to transport it down a concentration gradient, but they don't need to be co-transported with sodium. The second type of carrier are the sodium-dependent glucose carriers or monosaccharide carriers. We have two major sodium type carriers in the body. So here in the intestine, what happens is you will have co-transport of glucose and galactose going through this sodium-dependent carrier. And of course, this is also ATP dependent because we have to power the active transport of sodium out of the cell to make a low concentration of sodium inside the cell, so sodium can move down its concentration from the lumen into the cell, and then by secondary co-transport, transport monosaccharides in. There's another, just a GLUT carrier that carries fructose in down its concentration gradient. And then on the basolateral membrane, what do we mean by that? This is the base of the cell over here. This is the lateral side, this is the lateral side. So this is the luminal side next to the lumen. So on the basolateral membrane, we can just move these monosaccharides down their concentration gradient. So they're not sodium dependent in this step, they're not co-transported. That moves them into the intestinal fluid, it goes into the capillary blood, in the villi, and then transports it to the liver first and then the rest of the body. So this is essentially transportation and absorption. Proteins, of course, are made of amino acids with peptide bonds, and we begin protein digestion in the stomach with pepsin, which has been activated from pepsinogen by the hydrochloric acid in the stomach. This basically chops proteins into peptides, shorter amino acid fragments, and these peptides are then shortened even further. Some of these are by the proteolytic enzymes, which come from the pancreas into the small intestine. And then as you get even further, you can have carboxypeptidases and aminopeptidases, which clip off on the two ends. Amino acids are co-transported either by hydrogen ions or by sodium ions into the cell. And again, there's some active transport of sodium, which helps lower the sodium concentration inside the cell. And then we have this movement by amino acid transporters into the blood. The byproducts, the end products of both carbohydrates, the monosaccharides, and the amino acids of proteins both go into the blood. The reason we mention that is most of the fat monomers are going to go into the lymph node, and so there's a little bit of difference there. Both carbohydrates and proteins are polar, water soluble. And so their enzymes can interact with them very easily because the enzymes are proteins, and so they're polar as well. The digestion of lipids, fats in particular, is a little bit different because fats, of course, are non-polar. So what happens is the lipase is the enzyme from the pancreas, which wants to digest the fatty acids, the triglycerides, the fats, into free fatty acids and monoglycerides. The lipase wants to digest it, break the bonds. But the problem is because when the fat gets into the intestinal lumen, the fat all clumps together and the watery luminal contents are around it. So lipase can only work on the periphery of this large fat globule, which isn't very efficient. So the bile salts come in and they emulsify, they spread the fat into small droplets, which increases the surface area so the lipase can now work all over the place and digest the fat much more efficiently. So that's what's happening here. Emulsification is familiar to us outside of the body. If we have a fatty substance on our plate, cheese or whatever, and we want to wash it off with the water, it doesn't wash off too easily, but we add a little soap, a little detergent, and it breaks up the fat. It emulsifies the fat, and we can wash it more easily. So the soap is an emulsifier. Well, the biosalts are acting as an emulsifier to break those fat droplets up. So we take a big fat globule, we add the biosalts, and we make these little fat droplets. And now the lipase, which does the digesting, has all this increased surface area to digest the triglycerides down. As it does that, we have these little structures called micelles. This is a triglyceride. It has a glycerol backbone, the 3-carbon backbone, and then 3 fatty acids, 1, 2, 3. That's a fat, triglyceride. The lipase can clip off usually 2 or sometimes 3 of those fatty acids to make free fatty acids, as shown here, and monoglycerides. And they're in this little clump with biosalts around them called micelles. In that form, the monoglycerides and the free fatty acids get absorbed from the lumen into these intestinal epithelial lining cells. So now they've been absorbed. They've been emulsified and then digested and then absorbed. But what happens to them once they get into the cell? Interestingly enough, we go backwards. We put the fatty acids and the monoglycerides back into triglycerides. But then we put a protein coat around them. This protein coat is added in the Golgi apparatus. And now we have a protein coat around the triglyceride fat, and we call that structure a chylomicron. So the protein is encasing or coating the triglycerides, the fat, and it gets absorbed from the basolateral side of the epithelial cell, but it doesn't go into the blood, it goes into the lymph fluid. The lacteals are the lymph vessels in the villi. Now, if it's small enough, fatty acid, like less than 10 carbons, some of it can go in the blood, but most of it, it's bigger, is going to go into the lymph fluid of the lacteals. Eventually it'll go into the blood because the lymph eventually drains into the blood, but it's absorbed into the lymph fluid and not to the blood directly. This shows you this process again and going into the lacteals. Do we feel okay on this? Pretty okay. Vitamins, if they're fat soluble, they'll be sort of absorbed in this mechanism. If they're water soluble, they can just absorb by diffusion. They're water soluble. But vitamin B12 is a little special. It has to have intrinsic factor from the parietal cells of the stomach, go into the lumen of the stomach, go down to the small intestine, and then aid its absorption, primarily from the ileum of the small intestine. Other waters and minerals just diffuse. Finally, this is just as simply, not to memorize, but if you need a reference as to what's doing what, this table from your book gives you a summary of all the different areas and what they do. A little bit on just some, not all, of the diseases that can affect the gastrointestinal tract. We know we have ulcers. Ulcers are where you can get a little depression, indentation along the mucosa of the digestive tract. If it's a ulcer in the stomach, we can call it a gastric ulcer or a peptic ulcer sometimes. If it's an ulcer in the duodenum, we can call it a duodenal ulcer, for instance. If the ulcer gets too deep where it infects the vasculature, the blood vessels, we can call it a bleeding ulcer. That's more serious. You're losing blood in that case. A lot of ulcers have been associated with a bacterium called H. pylori. You've probably heard about that. And so that can be a factor. And stress and excessive acid probably also play a role in forming ulcers. Vomiting can be caused by many physiological reasons. We could have eaten something that didn't agree with us. We could have the flu or some kind of either viral or bacterial infection that makes us nauseous and vomit. We could have very low blood pressure. We could have a head injury like a concussion. We could have ridden the teacups at Disneyland, something. A lot of things make us want to vomit. This is basically reverse peristalsis. The most logical explanation is vomiting is a mechanism to help us get rid of something that could be hurting us, but it does happen in other situations as well. Gallstones, when certain substances precipitate out of the bile, we can get gallstones, which can eventually block the release of the bile into the duodenum from the gallbladder. If they get too bad, you might have to remove the gallbladder. You can live without a gallbladder. You just would tend to want to eat smaller meals, more frequent meals. And if it's a high-fat meal, don't eat too much fat at any given time. The bile is still made by the liver, but you don't have that larger dose from the gallbladder if you don't have a gallbladder. Lactose intolerance, imagine you've heard of. Basically, lactase is the disaccharidase that is going to catalyze the hydrolysis of lactose into its monosaccharides. And if you either have a mutation or don't have the gene to make proper amounts of lactase, you won't be able to digest lactose. So we call it lactose intolerance. Basically, the lactose is the sugar that's found in dairy products like milk and cheese and ice cream, certain types and all that. And so it can give you diarrhea and gas because you're not digesting this, so more water stays in your lumen and gives you those kind of problems. Inflammatory bowel disease, still trying to learn about this. For some reason, and you know that bowel just can refer to intestine, small bowel is a small intestine, large bowel is a large intestine. For some reason that gets irritated and you have to go to the bathroom more frequently, have bowel movements more frequently. And stress seems to be part of that. Some people seem to have a genetic predisposition for that. There might even be some kind of mind-intestinal connection that's causing that. So sometimes they give antidepressants to treat inflammatory bowel disease, for instance. Often there are dietary triggers that make it worse, so you try to avoid those dietary triggers. Celiac disease is an autoimmune disease where if you have gluten in your diet, then it triggers the formation of antibodies that attack your villi and your microvilli. So your villi is supposed to be these finger-like projections, they get flattened. And so your absorption is compromised. And so the only real treatment is not to eat gluten anymore, so that you don't trigger those antibodies. And of course, constipation means the motility is going too slow, so the bowels, the feces are getting too hard. Diarrhea is the motility is going too fast, so it's too watery in that case. this gives you a little bit more information about ulcers. Oh, let's see. We're out of time, aren't we? This just shows you an endoscopy where you're putting a camera down to look from the proximal side. Colonoscopy would be from the opposite side. These are just things that make us hungry and things that make us feel full. So maybe look those over. And this is just the dietary recommendations. The last couple slides, you could just kind of look over and it's not too complicated. All right, so tomorrow we start with the renal or urinary system. Get a good start, and we have that audio lecture on Canvas for Friday. Then after break, we come back and we have a little sprint to the finish. We finish up the renal, we do reproductive in those 4 days. Then we have the final test. Test week on Wednesday, test week. We have a couple, 2 or 3 more Connect assignments due. We have 2 reproductive case studies that we'll be doing. We have the book report. We have the reproductive video. And this one, I think that's, yeah.
Do you know what day the final is gonna be?
It's on Wednesday at 12:45 test week. So when we come back, we have a week of dead week and then we